listeners. Yamina here. Welcome to the Dr. GPCR podcast. Today's guest is a scientist who didn't plan to be one. Dr. Jens Carlsson grew up in a small village in Sweden where he had never even met a PhD holder. Now, he leads a powerhouse team at Uppsala University combining computational chemistry and medicinal chemistry to decode how GPCRs recognize, respond, and reshape biology. But the journey wasn't linear. From a lousy experimentalist to a global expert in receptor modeling, Jens's story is a masterclass in chasing curiosity and turning failure into focus. We'll unpack the difference between explaining and predicting, and what happens when models meet molecules, and how a cold email led him straight into the heart of GPCR discovery. Theory's Corner gives you the framework top drug hunters rely on, distilled into bite-sized lessons by Dr. Terry Kinakin himself. Each session translates complex receptor pharmacology into action-ready insight. Training drops every week, plus live Q&As every month. Explore it at terrykinakin.com. This month, we are excited to spotlight Revity's pH-Sense reagents in the Dr. GPCR ecosystem. It's a breakthrough toolkit designed to make GPCR internalization studies simpler, faster, and more scalable. With 4 flexible formats, no-wash protocols, and TRF detection, pH-Sense gives researchers the ability to monitor internalization in real time, even at endogenous expression levels, without imaging. And a quick thank you to our collaborators at Celtaris Research. Their fluorescent ligands and real-time binding assays are enabling sharper GPCR discovery through chemical biology. All the links that I've mentioned are in the notes of the show. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR, and I'm very excited to be back in 2025, recording new podcast episodes just for you. This very early morning in January, I have the pleasure of having with us Dr. Jens Carlsson from the University of Uppsala. Jens, welcome to the podcast. I'm very excited to have you on.
Excited to be here.
Let's start at the beginning, because this podcast has been in the works for a long time. I do remember getting to know a little bit more about your research and making a mental note of, we have to send an invitation to the podcast. That took a while. But then I think scheduling this podcast also took a while. Better late than never. Let's start at the beginning. Could you please introduce yourself, tell us who you are, and what is it that you do?
All right. So I'm a professor of computational biochemistry at Uppsala University since about 2 years back. My group always starts from the structure. So we're interested in the structure of the receptor and see what we can learn from the structure. And it's often about interactions with ligands and how they modulate receptor function. We use lots of different techniques, such as molecular docking, molecular dynamic simulations, increasingly machine learning with protein structure prediction, and try to see how we can connect pharmacology to the actual structures with simulations. Since a few years back, I also have a medicinal chemist, so we make compounds that we design. And we try to answer questions such as, what is the activation mechanisms? Why do the receptors recognize this type of compound and not another? And try to design molecules with new properties that can be either chemical probes to understand function or starting points of a drug discovery project. Now I don't hear you, actually.
Of course not, because I muted myself while I was drinking a sip of my coffee. So I love it. Apologies. It's still early morning here. My question was more around, did you always know you wanted to be a scientist, and what led you to where you are today?
No, I did not know that. And I got into GPCRs very late. So in 1998, I moved from a small village in the south of Sweden to Uppsala to study to become an engineer. And at that point, I'm trying to remember, but I don't think I knew what a PhD was. Like I'd never met someone who had a PhD, and scientists were, in movies or comic books. So I didn't really think much about that. It was around the boom of biotech. So I'd read about the sheep dolly and engineered tomatoes. So I thought, okay, this is the future. So I went to a program called biotechnology engineering. They needed engineers that could work with biology. That hadn't really existed before.
Mm-hmm.
And during that time, I did some internships to try to understand what I wanted to do. And I realized that I'm a lousy experimentalist. So I did a summer internship and I'm purifying proteins. And after the internship, I wanted a recommendation letter, and the professor wrote it down and sent it to me. And it didn't say almost anything about the actual protein purification. Instead, it said I was gifted in the modeling part. And I thought, did I do modeling? In my spare time, I was sitting looking at structures that they had solved. But what I think it meant was that I was a lousy experimentalist. But I read it as if I was a gifted modeler. So I thought, this is my thing. So let's continue along that path. And at the end of my master's, I asked the professor who was doing molecular modeling to give me 3 names of professors in California, because I wanted to do my thesis in California. I still have that note somewhere. And 2 of those actually got the Nobel Prize about 10 years later. And I emailed them, and one of them replied. I ended up at Scripps in San Diego, where I studied how proteins reacted to pH and how to treat water in simulations. So still nothing about GPCRs, but I realized there that the people working there, they were really cool. Like, I remember sitting in the coffee room, they were talking about all these different simulations, and I felt at home there. So I went back to Sweden, started my PhD, which was then on small molecule design. Like, how could we design ligands of proteins? As there were no structures of GPCRs, I never encountered them. I didn't know about them, anything actually about receptors. But working with the modeling, I felt that collaborating with the experimentalist was kind of like a waste of time. I saw that the students next to me who were protein crystallographers, they were just purifying proteins over and over again and failed over and over again. Whereas I, as a modeler, I just went to the PDB and I could get the beautiful structures, look at them, use them, without having to go through that experimental pain. And that has changed a lot now, but back then I was really focused on the computation. Then at the end, I realized somehow that there's a difference between to predict and explain. Simulations, people working on simulations spend a lot of time explaining. Like, we get the structure, it has this function, or it binds this compound, and they try to explain that. But fairly few of them were actually predicting things. And let's say you could explain what the weather was like today. Could that model explain what the weather would be like tomorrow? And I felt many of those would fail. They would not be able to predict the weather tomorrow. They didn't make things up. It was a theory. Could be right, could be wrong, but I needed to approach the experiments. So I started looking for a postdoc position, and if I added all the constraints I had, it needs to be someone who's a modeler that can do experiments, and the system has to be so simple that I would believe the computations will actually work. And when I added all these constraints, there was only one name in the world that I could go to, and that was Brian Scheuke at UCSF. Okay, so I emailed him, and I've seen he's been on the podcast, so and works on GPCRs, but at that time he didn't. So that's not the reason why I went there. But he had the setup that I thought that I needed to continue. So I went there, and very shortly after I joined his lab, he came to me and asked, like, hey, Jens, maybe you should work on GPCRs. And I thought, what's a GPCR? So I went back to Google the GPCR, and I found, okay, they seem to be an important protein. And why not? There seems to be a lot of excitement around this. And a colleague of mine was working on the beta-2 receptor, which had then just been determined. Everyone was so excited. So I thought, okay, let's try GPCRs. And since that day, it's been my main topic. So it was all just coincidence of some kind. So the first collaborator I had there in Brian's group was Ken Jacobson, who you also recently interviewed. And we worked on the A2A adenosine receptor. And it wasn't difficult to select target at that time, because there were only 3 structures, or 4 maybe: rhodopsin, beta-1, beta-2 adrenergic, and adenosine. So let's pick number 4. So I tried to identify ligands using virtual screening, and that went really well. And then I thought, yeah, okay, so we can predict the future. We can predict the outcome of experiments. So I then jumped to the dopamine receptors together with Brian Roth and Brian Schoike. And there we actually tried to predict the structure of the receptor, not use crystal structures. And later crystal structures were published, and they looked the same as our models. So I thought, hey, yeah, it works. Modeling can work. And that's how everything started.
I love it, and I think the pairing with Ken Jacobson was a good. I know that his lab, from our interview together, was very much focused on small molecules. So they have this plethora of molecules that you can then use as a starting point to get a sense as to how do those molecules that are known, for which we have experimental data, potentially interact with the adenosine receptor, and use that knowledge to then predict the future there.
A great mentor for me, and he's very open-minded. He actually does a lot of early modeling himself, and that's really unique about the GPCR field, that there's a close connection between experimentalists and modelers. I think that's what I felt when I was a PhD student, that there wasn't really a good collaboration. But in the GPCR field, there's so much opportunity for good collaboration.
Yeah, and I think, to be honest, I've always been in the GPCR field, and I always felt at home just because of that. The fact that people are very collaborative. It's very easy to get in touch with others, and then you talk to one person and said, oh, wait a minute, you should talk to that person. And then that's how beautiful collaborations start off, and allows you to navigate.
I remember I was on the email when Brian Schoike contacted Ken Jacobson. It was like 2 lines. It said, I heard the structure was out. Can we send you some ligands to test? And Ken replied, yes. There wasn't any negotiation or about anything. Just like, yeah, wow, that's really exciting.
That is fascinating. I really love it. You mentioned in the beginning that you wanted to move to California. What was let's say, the motivation there? In a sense, you restricted your geography, but at the same time, it's California, and there's fantastic universities and research labs there.
I think, it was more that I was looking for, the first time, I went there as a master's student, then it was the weather. So I wanted to go to California. And I had no idea then that all these great scientists were working there. But in the second case, I had decided on the lab I wanted to go to. Yeah. I wouldn't really have cared what the weather was like. But I think my family appreciated it. I had one daughter with me, and then 2 when I left. So we really loved living in California.
Lovely. Yeah, I was asking about the first time. Second time, it was Brian Schoike's lab and the team. Phenomenal. All right. So when you were completing your postdoc, did you ever think about not going into academia, maybe going into industry and using the skill that you had developed, or was it a clear cut? I'm finishing my postdoc and I'm looking for faculty positions.
Yeah, so I knew that I was going back to Sweden because for family reasons. And I also knew that I wanted to pursue an academic career. And I told myself that, okay, I'm going to run for as long as I have funding. I don't want to be a person, you run out of funding and you just struggle. So for as long as I enjoy it, and I have resources to do what I want to do. I will continue. And I've been lucky that has been the case. But I still, even now, sometimes people ask if you ever considered industry position. I still think today I haven't made up my mind. If I don't find myself comfortable anymore in academia, I could go to industry. Yeah, it'll be fun.
Absolutely. And the reason I was asking this also is because we talked previously, I think it was late last year in a different context. And I know that you have a consulting company, or you have a consulting arm of your lab. Maybe if you can talk to us a little bit about how that came to be, and where do you think that's going to go?
So I started this consultancy company because I repeatedly got requests by email from either individuals or companies who wanted advice or actually work to be carried out. And I didn't know what to answer. Like, how would I be able to do that? So then I started, after getting one of those requests again, I started the company, and that allowed me then to be part of advising a company that was designing ligands that I was experienced in. So, and I also met there was a lot of other people involved that were people that I really liked. So I really jumped on it because I got the chance to hang out with all these people that I admired very much. So that was nice. And now I have a few other things I'm doing now with the company, advising research groups. But the company doesn't do any actual computational chemistry. It's just can provide on strategy. But I hope in the future maybe we could do that. I need the academic work takes a lot of my time, so I don't have time. But maybe in the future.
Absolutely. The reason I was asking this is because, again, thinking about the audience, and I want people to get a sense of how did you get into your current academic position. But the fact that you're having currently an academic position doesn't mean that you're going to retire from this academic position. It's all about your preferences and where does research take you, but also knowing that you have other opportunities if you'd like to pursue them. That's very important. All right, so let's move on. Let's move on to the next, unless you had a comment there to add.
No, I think that I just try to grab opportunities when they come to me. So during the pandemic, for example, we worked on enzymes involved in SARS-CoV-2, identified very potent compound there. I just love molecules, like working with proteins and molecules. That's my driving force. So if I see an opportunity to do that, soluble proteins are great because you can determine a lot of structures. So all proteins have their pros and cons, but still everything comes back to the receptors at the end.
Yeah. I always tell people that in my mind the receptor has its own personality, and it'll tell you what you need to know, or if you're using the right conditions to tell you how it likes, to bind ligands. Is there calcium? Is there water or no water in there? And how that structure influence the function of the protein and of the GPCR.
And they all have the same structure. When I came back I was in Stockholm first before I came back to Uppsala. Then there was a professor there that told me that, these GPCRs, the structure is known. It's 7 helices. And then I realized, okay, maybe we have a little bit different perspective, because I care where every atom is. And these 800 receptors can bind so many things using that same scaffold. It's very fascinating to me. So I think that will keep me busy until I retire.
I'm sure. I recorded earlier this week another podcast episode, and with my guest, we were talking exactly about this aspect of the fact that there's 800 receptors, including the olfactory ones. There's a limited number of G proteins, even more limited number of G protein subunits, yet the receptors have this amazing ability to signal and to control so many signaling pathways and physiological outcomes. And it's fascinating. As you said, they're all 7 transmembrane domain proteins, yet they all do different things.
Yeah. Yeah, I tell the undergrads that they are a GPCR, like what they think, what they see, what they do. You're a GPCR, basically.
Yeah, I typically introduce GPCRs to people who don't know it as GPCR. You can see, smell, and taste thanks to GPCRs because that's the easiest way to represent it. But I like you are a GPCR analogy. All right, so you mentioned in the beginning that when you started working on GPCRs, there were just a few that had structures. Is there, out of that, or since then, do you have a favorite one that you kind of had?
No, I don't have a favorite GPCR, and I try really hard not to have one. It's difficult. The adenosine receptor was kind of my first love. You know, it was a structure. There were many it was interesting therapeutically for Parkinson at that time, and we published several studies together with Ken Jacobson and other groups on that receptor. But I couldn't stay there. I felt that that you have to, each GPCR has a different challenge, and often there's a structure that triggers a question for our group, like a new structure, or there is a specific question that leads us to work on a specific receptor. So some receptors are simply not suitable for answering the question, and some structures, you have to move to different targets to answer different types of questions. And I think that really helps us also to get general conclusions that will be applicable to many different targets. But that said, I truly love going to conferences and talk to the person about this receptor, which is like an encyclopedia of information and knowledge that I can try to tease out and combine with some other knowledge to do something new.
Lovely. No, absolutely. I think it's great. You mentioned in the beginning that I really loved your story about the recommendation letter that said that you're a great modeler. I love the attitude as well to take it as I'm a great modeler. And you mentioned that now in your group you do also have experimentalists. Can you walk us through a little bit more about on how is your group divided now, and what kind of experimentalists do you typically work with in the lab?
So the project starts with a question. It could be, can we design agonists of this receptor, or can we distinguish between agonists and antagonists? Then we choose a receptor target and try to connect to a pharmacology group that could test the prediction. Because since we want to predict the outcome of experiments, the story has to end with an experiment. So first we can develop a method, a technique, and then we want to try it. Then early in the project, when we purchase compounds, we usually buy them from commercial libraries. And as you probably know now, there are billions of molecules that you can buy online from NMN databases. And we can screen in the lab probably a couple of billion molecules in a couple of weeks on the supercomputer centers. So that's the fastest way to get going. Then every project at some point hits the wall. That there are no commercial compounds that are interesting anymore. And at that point, our chemist, we have one chemist and about 10 computational chemists, and then he comes in and makes critical compounds that we believe must be made but cannot be purchased. I don't have any pharmacology. I tried that a little bit inside the group, but I found it difficult. And whereas chemistry, where I would say it's relatively easy to convince a pharmacologist, maybe you can try our compounds. If they are experts in that receptor, they're maybe running that receptor assay every week. It's much more difficult to convince a chemist to make the compounds. They've been disappointed by computational chemists. They've been disappointed by our predictions so many times that they often they don't even answer your emails. Well, so then I realized I have to have a chemist if I want to make molecules, and sometimes we want to make really design libraries and make very specific molecules, and then I need the chemist. But I'm not an expert in medicinal chemistry, so I have a colleague helping me to supervise a fantastic researcher that I have in the lab.
That's fantastic. And I think from a pharmacology perspective, you have you have so many choices because there's so many labs that work on so many receptors in general in the field. And going back to the fact that the GPCR field in general is very generous, you could do just as Brian did. We have, do you want to test these? And then Ken responding yes, type of situations. But I can see Brian sending that email. I've not only recorded the podcast with him, but met him in person in 2023 at the Gordon Conference. 23? Yes, 23, in Switzerland.
And my experience there really changed how I work. That I realized that to accomplish the goals I had to collaborate. And sometimes it's painful, if you can't, with an international groups, you often cannot get a joint grant. Like you have to get funding in each country. That can be difficult. Maybe the collaborator gets funding for a different project than I got. And the pandemic was very challenging. A lot of labs closed down. So collaborating is very challenging, but much more fruitful than just focusing on your own thing. You learn so much about their perspectives on what is important, what do they actually want to find, what type of compounds, and why is it important. So it's been very important for me to interact with the community.
Yeah, and they can tell you what your molecule does to the receptor, and how does that molecule influence the receptor, and what does it do to it, and more information. Because sometimes I also work with medicinal chemists, I work with computational chemistry people, and at the end of the day, it's the biology that matters, and not that the other fields don't, but one makes the molecule, the other one predicts it. Without the biology, without testing the molecule on the receptor, you won't know, you won't learn. And having the right assay that's reproducible, that gives you the curve, and be able to look at that curve and say, wow, okay, so this is what's going on, this is how the data looks like, is valuable information that allows you to then go back through that design cycle and tweak your molecules to get a sense of, okay, what buttons or what residues do we need to interact with on the protein in order to create the desired pharmacology.
And when I talk to industry about my students who have reached industry, that's one of the things they tell me is that they can interact with the chemist and the experimentalist and interpret the data, because you really need to be able to do that. Coming from a mostly computational background, that takes a lot of time actually. But I realize I need to understand the experiments also in order to really use the data. So they learn from us a little bit sometimes, and we learn from them.
Absolutely. And I think it's also about developing that common language and be able to communicate and have productive conversations with each other, where let's say the computational teams understand what the assay is. They don't have to know how to run the assay, but understand what the output is. And then also from the biologist to understand what is the perspective, where does the computational chemist, where does the medicinal chemist come from, and how can I as a biologist provide them the information they need in order to go ahead and improve on those molecules.
Yeah, and I think a very important role is to be able to explain what your model can and cannot do. So everyone wants the answer to a very complicated question, a very difficult question. And many times, maybe the model cannot explain that. So sometimes we have to just answer no. That is not within the accuracy of our method. Like, is this compound twofold better than another? Then I would say, nor the experiment or the computation has the resolution to tell you in many cases. So that's a most important role, is to tell you what, maybe not what can be done, but what cannot be done. It can sometimes be more important.
I agree. And I think it goes same way for the biologist and the experimentalist as well, because assay sensitivity, detection limit, all of these things also matter. And until you have an optimized enough molecule, you can run the experiment 4 times and get a variable, piece of data each time. And then oftentimes you need to be able to explain, well, actually it's the molecule that's not optimized, and we're in that lower assay window that doesn't allow you to very nicely capture a beautiful dose response.
Yeah, exactly. It can depend on solubility or how the sample has been prepared, and then the students, my students, can say, we can't trust this guy. He gets sometimes 50 micromolar, sometimes he gets 10 micromolar. Well, at this stage it's the same value.
Exactly. No, I agree, and I learned that in kind of in the hard way, because I've spent a lot of time explaining, okay, this is the assay, and yes, whether the Emax is 70 or 100 with an unoptimized molecule, that's pretty much the same thing at that point. Wonderful. All right, so let's go into the last segment of our conversation. And as I mentioned before we started recording, is the goal with the podcast in general is to inspire junior scientists, but at the same time allow them to get a sense of who you are and what do you do, and last but not least, get some advice. So if you had to give advice to junior scientists aspiring to contribute to the field, what would that be?
So on a general level, I think you need to pick a problem that you can't stop thinking about. So if you're not genuinely interested in what you do and just maybe picked it because it's a hot topic or it seems important, then you won't have the energy to complete the project or pursue it, because you will have so much failure. Like, we fail all the time. Most of the time, the experiments fail, the calculations fail. So you need to be able to wake up in the morning and say, hey, wow, that problem. Like, when I was a PhD student, I felt every time I woke up, the first thing I was thinking, what with my calculations, how did I end up from yesterday? And I think find a problem that makes you really excited. Then I think mentors is really important. I've had fantastic mentors, and you should ask a lot of questions. Should be afraid to approach people, ask them what they think. Then you find the ones which give you valuable information, but don't always. I don't always do what they tell me. Like, you have to think about why are they saying something, and then make your own decision. So when people say, oh, that's not an interesting problem, or that will never work, that's like a trigger for me. You tell me something won't work, then I'll say, okay, we're going to work on that. And so find people that can be your mentor, and be happy about the small successes. That would be my third advice. That you need to be able to be happy about a single small experiment working, because if you're only happy when you get a big paper published or you get this big grant or get promoted, then you won't have much happiness. So be happy about the small things that excel the project.
I love that advice. I very much love the mentoring part, but also about the small successes, because I feel like these questions or these problems are so complex. We may not be asking the right questions, or we're asking the questions too soon, either because we don't have the tools or when we don't have the knowledge to answer those questions. And very quickly that interest and that drive can, dissipate. So being able to celebrate small steps forward is very important to keep that interest. And when you wake up in the morning, you know that, okay, this is the question, so how does that work? And that motivates you to go back to the lab and run those calculations or run those experiments. I really love it. Thank you. All right. Top 3 aha moments that shaped your trajectory as a scientist.
I think the moment I realized that I was useless in the lab, that was an aha. I knew that for a long time, but I had to somehow try before it was settled question. So that really set the direction for what I wanted to do. So I was very focused from that point on learning programming and molecular modeling techniques. And that's also how I met my PhD supervisor. And so that was one. Then the second aha moment was during my postdoctoral research, finding how fruitful collaboration can be. That really changed, and that I have sort of the the possibility and an ability to somehow interact with other people, form projects, and carry out those together. Whereas I initially viewed science as someone sitting alone in a room doing simulations. That's a little bit how I was raised somehow, that you worked on a problem. Maybe that's how I saw it in the cartoons. There was this crazy scientist alone in the basement. That's how I viewed science. But now it's much more collaborative.
Yeah, you mentioned. Please go ahead. I'll comment afterwards. You mentioned the cartoon and the crazy scientist, and I had a mental image that came into mind. Please.
I don't know. Yeah, I think those are two things. I realized collaboration would be very important for me, and that I'm not an experimentalist. And also what I talked about, this difference between explain and predict. Okay, scientists think they can explain all things, but can they predict it? That's something that I still struggle. Every time we do a project, I think, okay, did we predict this or did we explain this? I really want to do perspective science, that we can use these models. We need to prove that the computational methods can guide drug discovery, not explain what happened.
Yeah. All right, two comments. One, the scientist in the comic book. And you're right, I think you watch movies or you look at comic books and the scientist is sitting somewhere in a basement in the dark, hair all over the place, and it's that lonely type of work where in reality it's not at all. And it cannot be. It cannot be that in order to make progress.
At least maybe there was a time where you can sit at your desk with pen and paper, a few individuals that could do that, but it's so much more fun and moves things forward so quickly if we work together.
Agreed. And then you said, predict and explain. I think with the coming up of machine learning, the explain part was important in order to be able then to predict. But now we have enough data explained to try and predict.
I think it's super exciting what's happening now in AI structure prediction. We have several projects, one published where we use AlphaFold models to identify ligands. So that's definitely super exciting to see both what you can do on small molecule design with AI methods and the structure prediction. But the rule remains the same. They have to be able to predict the weather tomorrow. And we still don't know. I've seen some amazing things with AI, prediction of protein or receptor peptide complexes, which AlphaFold cannot have access to. And we started modeling this complex structure in the lab. And when I saw the AlphaFold structure, I said, yeah, this is it. It must be right. I just did what I usually do, read the literature, find mutagenesis. But AlphaFold, it just 1 second, it beat me. And I saw later the structure came out and it was right.
Wow.
So that really shocked me. But then I ran into the lab and said, no, we have to try this out. Then it didn't work as well when I tried it on another case. But it's a true breakthrough.
Wonderful.
We just learn how to do it.
Yeah, I think so too. And I think it's also about how much information is there that we can start explaining what's the starting point. in order to then start predicting. And I love the analogy about the weather, because nowadays the weather is very difficult to predict, almost as difficult as predicting GPCR structures.
Yeah, and for small molecules, I think there is a data problem. So what enabled the structure prediction breakthrough was a lot of sequences. And for small molecules, we don't have that information. So some new type of data is required to really make a major advance there. And we had some experiences with the new AlphaFold 3, and yeah, as expected, maybe it's not working as well as we had hoped.
It's a matter of time.
Yeah, we'll see.
All right, last but not least, this is, I think, the toughest question of them all. What should we title this episode?
I have no idea.
I typically, it's in the questionnaire for two reasons. One, I'm very bad at naming episodes, and number two, we cannot pick a title ahead of time because we don't know how the conversation will go. And so I decided to involve our guests and try and find an episode title. I think it's a tough one because we talked about many different things, but it was mainly I liked very much the weather analogy and predicting versus explaining. So I think that's that. On a more, personal front on your end is really the modeler versus the experimentalist, or the experimentalist turned modeler type of vibe that came through. What do you think?
I don't know. Maybe the explain versus there's a difference between explain and predict.
Yeah.
Okay. I don't know what the other titles look like.
Well, it really depends. I've had, and now I'm actually, these episodes will have come out by then. So we have research the gene that skipped a generation, computational chemistry of olfactory GPCRs, non-canonical path to a PhD via the opioid epidemic. We have something around from curiosity to innovation. So it's really, we can be as creative as we want to. Predict, not explain.
Yes.
I think that kind of goes through the spirit of our conversation.
I think so too.
Wonderful. Jens, thank you so much for your time. I really appreciate. I had a really fun time getting to know you more.
Fun too. I appreciate it very much that I got this opportunity.
Thank you so much. Don't go anywhere.
Thank you to Dr. Jens Carlsson for such a thoughtful conversation, equal parts human and technical. From failed protein purification to shaping how we model receptor behavior, his story reminds us that clarity comes through persistence. Here's what you can do next. Share this episode with a colleague, subscribe to the Dr. GPCR podcast, and leave us a review. It helps others find the podcast. Want to level up your GPCR know-how? Join Terry's Corner for weekly pharmacology lessons and exclusive live Q&As with Dr. Kinakin himself. Start learning today at terrykinakin.com. At Dr. GPCR, our mission is to advance receptor science through education, community, and by spotlighting tools that move the field forward. And Revity's pH Sense is just that, a real solution to persistent internalization bottlenecks. With pH Sense, you can finally track GPCR internalization in real time, even at physiological expression levels, using a simple plate-based format. The reagents are compatible with HTRF readers. It's validated on GLP-1R and mu-opioid receptors, compatible with all HTRF readers. You can explore it today by visiting the link down in the description of this episode. And special thanks to Celtaris Research for supporting receptor science through innovative non-radioactive ligand tools. I also want to take a moment to thank the entire Dr. GPCR team for helping making this episode possible, because receptors don't work alone and neither should you. Until next time, stay safe.